Now, guys, it's part 3 of chapter number 4, active transport of substances through the cell membranes. What is actually the active transport? Active transport is, for example, active transport is the transport of a substance against the concentration gradient with the use of energy. This is called the active transport. Active transport, we have two types of active transport. One is the primary active transport and another is secondary active transport. The primary active transport, for example, uh, if substance is less in on the outside of the cell membrane and it is more on the inside, and still this carrier protein carry this substance from low concentration to a high concentration with the use of energy. This carrier protein contain enzymatic activity, and with the help of this enzymatic activity it convert the adenosine triphosphate to adenosine diphosphate and organic phosphate, and the energy which is released is used by this carrier protein to carry the substance against its concentration gradient. We will further discuss the examples. Now let's take a concept. Now the secondary active transport. The secondary active transport is further are of two types: co-transport and counter transport. The co-transport. Let's first discuss the co-transport. For example, this is the cell membrane. This is the carrier protein. This is substance A and substance B on the outside of the cell, and this is substance A and B on the inside. The substance A is more on the outside and the substance B is more on the inside. So, the substance A, due to its natural affinity and due to its concentration gradient, will naturally attach to this carrier protein, and this carrier protein has also another attachment point, another receptor, which must be first attached to the substance B. So, the structure is organized in such a way that it will not carry the substance A until the substance B is attached to this carrier protein. So this substance B take advantage of the concentration of substance A, and then through the carrier protein both of them enter to the cell. So this was what? This is the example of the co-transport of secondary active transport. Now we will discuss the counter transport. This is, for example, the cell membrane, the carrier protein, substance A, substance B. In this example, the substance A is more on the outside, and the substance B is also more on the outside. But the substance A and B on the inside is in small concentration. So what will happen? For example, if the substance A has to be carried to the inside, then the substance A will attach to its receptors on the outside of this carrier protein. But this carrier protein has such a conformation, and this protein has such a structure that it will not carry the substance A until the substance B is attached to the receptor on the inside of this carrier protein. So the substance B will attach to this carrier protein, and the substance B is in less concentration in the inside and it is more on the outside. So the substance B will attach to the inside and the substance A will attach to the outside. So it will again use the concentration gradient, the energy of the concentration gradient of the substance A to carry in the opposite direction to the outside of the cell. So the substance B is carried to the outside and the substance A is carried to the inside. This is called counter transport. In this example of the co-transport, the substance A was more and the substance B is less. But in the counter transport, both is more on the outside, and the substance B is carried against its the concentration gradient, and but uses the energy of substance A. And in this example, substance B also uses the energy of substance A. So the primary active transport uses the ATP to carry out its job. But the secondary active transport is actually uses the energy of a secondary compound to carry out its job. Is a co-transport or counter transport. Co-transport, when it is co-transported with another compound, then it is called the co-transport. When it is transported in the opposite direction to transported substance, then it is called the counter transport. These were the example of counter echo transport. Now guys, it was the best I could do. Now we will discuss the examples of the primary active transport. Sodium potassium pump transports sodium ions out of the cell and potassium ion into the cell. For example, this is the sodium potassium pump. This big unit is the alpha unit and the small unit is the beta unit. The alpha unit has a molecular weight of 1 lakh and the beta unit has a molecular weight of 55,000. So the function of the beta unit is not understood, but according to the Guyton, that the beta unit maybe functions to anchor the alpha unit, and the alpha unit is the main functional unit of this sodium potassium pump. Now it has it has a receptor for the potassium on the outside of this carrier protein and receptor for the sodium on the inside. If it attached to potassium on the outside receptor, it must attach 3 sodium on the inside in order to carry the transport functions. Unless and until this transport protein will not transport potassium to the inside until and unless the 3 sodiums are attached to the inside of this channel. So when the 3 sodiums are attached to the inside of this channel and the potassium is also attached, then the ATPase function of this enzyme activated and converted the ATP to ADP and inorganic phosphate, and this energy is used. This ATPase, the breakdowning of ATP and enzymatic function of this protein will activated when the potassium is attached and to the outside and the sodium is attached to the inside. Then this ATPase enzymatic function will activated and it will perform its job to carry its 2 potassium to the inside and 3 sodium to the outside. And in this way, it's maintained the concentration gradient on the 2 sides of the cell membrane for sodium and potassium. Now this is done. Now we will discuss the sodium potassium pump is important for controlling cell volume. For example, how the sodium potassium pump control the cell volume. The sodium potassium pump will control the cell volume if for example, if the volume of a cell is increases due to the osmosis of water, then the sodium potassium pump will activate and it will carry the sodium ion to the outside of the cell. When the sodium ion is carried to the outside, then due to the ionic differences, the osmosis of water will occur from the inside to the outside and the cell volume will be normalized. And if the volume is decreases, then vice versa will occur. And in this way, the sodium potassium pump control the cell volume. Now, electrogenic nature of the sodium potassium pump. What it means, the electrogenic nature means that it produces the voltage across the cell membrane. This is called the electrogenic nature of the sodium. For example, the sodium potassium pump carries 2 potassium to the inside and carries 3 sodium ions to the outside. So there is a deficit of one positive ions. For every cycle of transport, there is coming 1 ion deficit inside the cell membrane. 2 positive ions are coming inside and 3 positive ions are going to the outside. So the net charge of the inside will become negative. So this is the electrogenic nature of the sodium potassium pump. Now the primary active transport of calcium ions. This is another example of the primary active transport, that most of the body cells contain the calcium pump. This is the reason why the calcium is high in the on the outside of the cell and in small concentration in the inside. This is due to the calcium pump. So most of the body cells contain calcium pump on 2 locations. The one location is on the surface of the cell membrane and the other location is on the intracellular organelles. For example, sarcoplasmic reticulum and mitochondria contain calcium pump. And in this way, the calcium is maintained inside the cell. If the calcium exceeds the normal value inside the cell, then the calcium pump on the cell membrane and on the sarcoplasmic reticulum and mitochondria will activate. The calcium pump on the cell membrane will carry the calcium to the outside, and the calcium pump on the sarcoplasmic reticulum and mitochondria will carry calcium to the inside of these intracellular organelles. So in this way, the calcium are maintained inside and the outside of the cells. Now, primary ectotransport of hydrogen ion. Hydrogen, this is another example how the hydrogen ion is transported. Hydrogen ion is a, for example, in, in the proximal convoluted tubule of the kidneys and the intercalated cells in the stomach. In these location, in the stomach, in the axanthic glands, in the axanthic glands of the stomach, the hydrogen is carried to the lumen of this gland with the help of the hydrogen pump through the primary ectotransport. And in the proximal convoluted tubule, the hydrogen is also carried to the tubules with the help of hydrogen pump. And intercalated cell also uses primary ectotransport to carry the hydrogen into the lumen of kidney tubules. These were the location where the hydrogens are pumping uh, out of this region with the help of primary ectotransport. Now, energetics of primary ectotransport. What it means? That it means that the energy which is utilized by the primary ectotransport is directly proportional to the logarithmic concentration of that substance. For example, they have given an example: if we increase the substance from 10-fold to 100-fold, then the energy will be utilized will be double. If we concentrate this 10-fold substance into 1000-fold, so if we concentrate the substance 1000-fold, the energy utilized will be 3 times. For example, thus in terms of calories, the amount of energy required to concentrate 1 osmol of a substance is 10-fold is about 1400 calorie. Where to concentrate it to 100-fold, then 2800 calories energy are required. So this was about the energetics of the primary ectotransport. Now, secondary ectotransport, co-transport and counter transport. We have studied the mechanism of the co-transport and counter transport, how it occurs. Now, we will discuss the co-transport of glucose and amino acid along with the sodium ion, which are the examples of the secondary ectotransport, co-transport and counter transport. Now, I have discussed in detail the mechanism of the primary ectotransport, the mechanism of the secondary ecto co-transport, and the mechanism of the secondary ecto counter transport. So I have explained the mechanism. Now we will take a look over the examples that and we will apply these mechanism over the applications of the counter co-transport. Now let's go, for example, the co-transport of glucose and amino acid along with the sodium ion. Let's take a look over the diagram. This is the glucose, the co-transport of the glucose, how the glucose is co-transported. So this is a carrier protein which carries the sodium ion. The sodium ion is more on this side. The sodium ion is more on the outside and is in less and small concentration on the inside. So the sodium, due to its natural affinity, like in the co-transport mechanism, so the substance A is like the sodium. The sodium has the natural affinity to attach to this carrier protein because of its concentration gradient. But this carrier protein has a structure and configuration that it will not carry the sodium ion until it is attached to the glucose, and the glucose is carried against its concentration gradient. So in this way, the glucose is co-transported with the sodium, and this glucose uses the energy of sodium. This is why this is called the secondary ectotransport. These secondary ectotransport of the glucose, and the amino acid is also transported in this way. The amino acid will attach to this place and the sodium will attach to this place. These phenomena occur in the gastrointestinal tract. The absorption of glucose and amino acid occur by this way. The reabsorption of glucose and amino acid in the kidney tubule, in the proximal convoluted tubules, the absorption of glucose and amino acid occur in this way. And so these were the practical application of the co-transport of the glucose and amino acid. Sodium counter transport of calcium and hydrogen. We have discussed the mechanism of the counter transport and how the counter transport is occurring. Now we will discuss the practical application and examples of the counter transport. This is the sodium. For example, in this diagram, the carrier protein has attachment site for the sodium and one attachment for the calcium. So this protein will carry the sodium ion to the inside until it is attached. It will not carry the sodium until it is attached to the calcium. So when the sodium is attached to the outside, this carrier protein will carry the sodium ions if the carrier protein is also attached to the inside calcium. So in this way, the transport occurs in the opposite direction. The sodium is present in high concentration on the outside of the cell membrane, and the sodium is present in small concentration on the inside. The sodium uses its concentration gradient to come to the inside of the cell, but the calcium secondarily uses the energy of the sodium and travel in the opposite direction. This is the counter transport which is occur in most of the body cell. Now the sodium hydrogen counter transport. The sodium is attached to the outside, like this is the same, and the hydrogen is attached to the inside receptor. These both will be carried in the opposite direction um, with the help of this carrier protein, and this carrier protein will carry the sodium to the inside only if these carrier proteins are attached to the inside molecule which has to be carried. So this counter transport of hydrogen primarily occur in this primary convoluted tubule, which are secreting the hydrogen ions to the tubule. Now these were the example of the calcium sodium counter transport of calcium and hydrogen. Now activetransport through the cellular sheets. Most of the body cells is attached to each other like in this manner. Most of the body cells is attached. This is one cell, this is another, and these are the adjacent cells. So most of these cells are attached to each other through the help of junctions, and they form a sheet-like structure. So these transport which we have discussed is actually not occurring through a single cell, but it occurring through this cellular sheet. For example, they have shown that the sodium ion entering, which is followed by the osmosis of water, and then they diffuse onto the opposite side of the cells. So the examples of the example where the activetransport occurs through the sheet, I have written one mnemonic, which is the REGIC. REGIC R mean the renal epithelium, which is also in a sheet, which is present in the form of sheet. The exocrine gland epithelium, gallbladder epithelium, intestinal epithelium, and the choroid plexus of the brain. The epithelium of the choroid plexus of the brain. So REGIC is the mnemonic through which the substances passes through the sheet instead of a cells. The substances passes through the sheet. These are this is the mnemonic for these regions. R mean renal epithelium, E exocrine gland epithelium, G gallbladder epithelium, I intestinal epithelium, and C epithelium of the choroid plexus of the brain. So REGIC, these are the areas through which the substance pass through its cellular sheets. This now guys, chapter 4 has done. Please guys, support my channel, like, subscribe and comment. I will be very thankful to you.